All right, welcome back, everyone. Today we're going to get into Chapter 10, and that is the start of a deeper dive into muscle tissue than we've ever gone before. And this lecture is going to be about skeletal muscle. So, what do we know about skeletal muscle so far? Well, we know it's excitable and it can contract. That causes movement. And it also resists movement, like how it resists gravity and it maintains body posture. We also just learned that muscles can prevent extreme movements, which helps protect bones and joints. And finally, if you think back to our brief lecture on muscle tissue when we covered skeletal muscle, I told you that skeletal muscle itself is voluntary muscle, meaning that it's under your conscious control. That allows you to do things like swallow, use the restroom, play sports, play an instrument, snap your fingers, etc. What else does skeletal muscle do? One example is that it helps maintain homeostasis, particularly through thermoregulation. Through shivering, you've noticed that after you've worked out, if you work out, your body temperature will increase because your muscles are contracting a lot. Uh, if you play a sport, or if you've just been shivering because of a cold day. So let's take a look here at skeletal muscle. I took this directly from the textbook. Every skeletal muscle is an organ, and as such, it's made up of various tissues. These tissues include the skeletal muscle fibers, of course, but also blood vessels, nerve fibers, and connective tissue. And so when you take a look at this image, I want you to notice that each skeletal muscle is going to have three different types or layers of connective tissue. We call these mesia. These mesia enclose the muscle, provide its structure, and compartmentalize the fibers within the muscle. Let's talk about all of them. The first is the epimysium, which is this sheath of dense, irregular connective tissue, and it allows a muscle to contract and move, and also maintain its structural integrity, and it also separates the muscle from other organs in the area, making it its own independent entity. The next mesia is the perimysium, and the perimysium basically organizes muscle fibers into bundles, each called a fascicle. We call this fascicular organization, and it's common in the muscles of the limbs. This allows the nervous system to trigger very specific movements by activating certain fascicles. And finally, within each fascicle, each muscle fiber is encased in a thin connective tissue layer of collagen and reticular fibers called the endomysium, which contains the extracellular fluid and nutrients that support the muscle fiber. Those nutrients are also, of course, supplied via the blood. Okay, so here we've zoomed in even further, and we've looked at individual skeletal muscle fiber. A skeletal muscle cell is long and cylindrical, and so they're called fibers. These cells are actually pretty big when you think about it, because some of them can get up to 11 inches long, which is crazy when you think about the size of a typical cell. During embryonic development, what you see is you got many of these myoblasts, which are just embryonic muscle cells that fuse together. Each myoblast has its own nucleus, and so adult muscle cell fibers are multinucleated. And each nucleus has a copy of the genome, which is good because that will allow for many nuclei to produce the large amount of proteins and enzymes that are going to be needed eventually for muscle contraction. The last thing I want to look at here is that some of the terminology that's associated with muscle fibers uses this Greek word sarco, which means flesh. The plasma membrane of muscle fibers is called the sarcolemma. The cytoplasm is the sarcoplasm, and the specialized smooth ER is called the sarcoplasmic reticulum, or SR. And we'll see soon that the functional unit of a skeletal muscle fiber is the sarcomere, which is a highly organized arrangement of the contractile and myofilaments that we talked about before when we looked at the organelles, actin and myosin. Now, before we leave this slide, I just want you to notice that the skeletal fibers appear to be striated, or meaning they look striped. And let's talk about why. The muscle fibers appear striated because of the arrangement of actin and myosin sequentially. Each packet of these microfilaments and their regulatory proteins, which are troponin and tropomyosin, is called a sarcomere. You talk about myofibrils, which are long filaments that run parallel to each other to form muscle fibers. Sarcomeres are bundled within those myofibrils. They run the entire length of the muscle fiber, and eventually it attaches to the sarcolemma at its end. As myofibrils contract, the entire muscle cell contracts. Because myofibrils are approximately 1.2 micrometers in diameter, hundreds to thousands of them, each that have thousands of sarcomeres, can be found inside just one muscle fiber. Each sarcomere is approximately 2 micrometers in length and has a cylinder-like arrangement. It's bordered by structures called Z discs, which you see right here as Z lines. Now, they're lines because this is a two-dimensional picture. They're Z discs when you look at the three-dimensional figure. It's bordered by those structures to which the actin microfilaments are anchored. Now, if you look at the actin microfilaments and its troponin tropomyosin complex right here, actin, troponin, and tropomyosin, sorry, keep hitting that button, actin, troponin, and tropomyosin is actually thinner. And so it's called because it's thinner, it's just called the thin filament of the sarcomere. And just vice versa, since myosin strands and their multiple heads have more mass and are thicker, we call those the thick filaments of the sarcomere. So I pulled two videos here that hopefully make it clearer how these work.
The repeated pattern of the sarcomere results from the arrangement within the myofibrils of two major proteins, actin and myosin.
There are the myosin filaments, the thicker, and the thinner are going to be the actin filaments.
Actin is a fibrous protein attached to the Z bands that are protein plates at each end of the sarcomere. The I band is a region of the sarcomere that contains only filaments of actin. The A band results from the presence of overlapping filaments of actin and myosin. The H zone represents the presence of only myosin. Let us now examine how a sarcomere undergoes contraction.
Yeah, let's take a look at that. That's the next slide I've got for you.
When a muscle contracts, the actin filaments within a sarcomere slide along the myosin filaments. This has the effect of pulling the Z bands closer together and shortening the sarcomere. This view presents a series of sarcomeres similar to the situation during contraction by a myofibril.
And so you can imagine thousands of these structures within each myofibril and multiple myofibrils in an entire muscle fiber that will just cause the entire contraction of the entire muscle. All right, so I think that's good enough. Let's move on to the next slide. Now, here we're talking about a diagram of what is called the Neuromuscular Junction. It's basically just a site where a motor neuron's terminal meets the muscle fiber. So some of this might start to get a little concerning, but it's pretty basic, and I'm going to try and break it down as best as I can. Basically, this is just how the nervous system controls the skeletal muscular system. It's how you can think, I want to snap my fingers, and then it happens. Every muscle fiber in your body needs to have a neuromuscular junction in order to receive these signals. If not, your skeletal muscle would not be able to move if they couldn't receive the messages from the neurons. Neurons and muscle cells can use their membrane potentials to generate electrical signals. If you remember from the previous lecture on neurons, a membrane potential is essentially an electrochemical gradient across a membrane. The differences in electrical gradients are maintained by movements of ions through protein channels. Both neurons and skeletal muscle cells are electrically excitable, meaning that they are able to generate action potentials. An action potential is a special type of electrical signal that can travel along a cell membrane as a wave. This allows a signal to be transmitted quickly and faithfully over long distances. Okay. For a skeletal muscle fiber to contract, its membrane must first be excited. The coupling of an electrical stimulus, which is the action potential, with a mechanical response, which is a muscle cell contraction, is what we're seeing right here. When the action potential runs down the neuron, okay, and reaches the axon terminal, it stimulates calcium to enter the axon terminal. Here we go. This is the axon terminal right here. This causes vesicles filled with neurotransmitter to undergo exocytosis, you see that right here, and release this neurotransmitter called acetylcholine into the synapse. That neurotransmitter, acetylcholine, binds to receptors that open up ion channels that causes the action potential to begin. Remember, this is a wave of electrical charge moving along a membrane. Once the action potential reaches the sarcoplasmic reticulum, it releases what it stores, which is calcium ions. It's the arrival of calcium ions in the sarcoplasm that initiates contraction of the muscle fiber by its contractile units, the sarcomeres. And we'll talk more about that in depth in the next lecture.